So now we're going to be moving on and talking more about DNA and the jobs of DNA. And there's two jobs for DNA. So remember that DNA stands for deoxyribonucleic acid. That's where DNA comes from, okay? You're going to need to know that. So the two jobs of DNA, the first job that we're going to be talking about is called DNA replication. And this is when DNA makes an exact copy of itself. And then the other job that DNA has is protein synthesis. Oops. And this is where we make proteins. Synthesis means to make. And what are we making? Proteins. So right now we're going to be focusing on this one right here, DNA replication, making an exact copy of DNA. So this is important because we need this every time that our cells divide. When our cells divide, we need to double all of the DNA inside of it in order for it to have two cells that have the same DNA, right? We want all of our cells to be the same. So let's draw a nucleotide. That our nucleotide has three parts, okay? 3 parts of a nucleotide. Okay, ready? We have our phosphate, PO4, our deoxyribose sugar that looks like a little house, and then a nitrogen base, right? Deoxyribose sugar. And how do I know that that's a sugar? Well, it ends in OSE, which means it must be a sugar. Remember that? All right. And then we have one of 4 nitrogen bases. So we have A, or we could have attached there instead of A, T, or C, or G. Now, we know that these shapes are not the actual shape of nitrogen bases. We just use these because they're a little bit easier to draw, and it's very clear what matches up with what. So the very first thing we need to do is I have only half of my DNA information here, right? And I need to do my base pairing matches. So I'm going to ask you, what matches with T? T matches with A. C matches with G. C matches with G. A matches with T. Okay, so here's our normal piece of DNA, okay, that we start with. And what's going to happen is we're going to have something come into this DNA and open it up, okay? So we need to open up our DNA so that we can read the bases that are inside and make copies. So what comes in here is an enzyme called helicase. Now, how do I know that this is an enzyme? Well, it ends in ASE. And anytime anything ends in ASE, that means it's an enzyme. And what this enzyme does is it unzips So helicase is going to come in and it's going to unzip our DNA and open it right on up. And how it does that is by breaking the hydrogen bonds. So the hydrogen bonds are broken by helicase. That enzyme. And now over here I need to again fill in my DNA on this side. So I'm just basically copying over from here and matching these up. A with T, G with C, T with A with T, C, A, G, T. Okay? So once it's opened up, now we can get in there and we can read these bases. And what gets in there and reads these bases and adds in the complementary strands is something called DNA polymerase. Okay? So I'm going to write that down. DNA polymerase. And do you think that this thing is an enzyme? Well, I'll tell you that it is, because it ends in ASE. And that means that it's an enzyme. So this is an enzyme, and the enzyme's job is to add DNA bases. So helicase opens up the DNA, and then DNA polymerase adds DNA bases to make new complementary strands. So we're going to do our same base pairing rules right here. Match them up again. So T matches with, C matches with, so that's one side, and then we need to add in the other side. So now you can see that we have two DNA strands that are exactly the same. We started with one, we opened it up, and now we're ending with two. Okay? The one that we wrote in has a special name, and these are called complementary strands. Complementary strands. So those are complementary strands. And then the strands that we started with that we didn't write in, the ones that were already written for us, those have a special name too, and those are called the template strands. And they're called the template strands because those are the strands that we used to make our complementary strands. So our template strands help us make our complementary strands. We start with the template. From the template, we make complementary strands.